goal is to produce a cell therapy that's both safe and effective for every single patient. But how do we know if we've been successful in doing this? Especially for autologous cell therapies where we know that the starting material will be different for every single production run. Ideally, we would like to be able to predict the clinical outcome of a therapy before administration to the patient, based on some set of attributes or properties of the product. Essentially, we just need some measure of the quality of the product that's predictive of the biological outcome and the clinical performance. We call these measures critical quality attributes, or CQAs, and a part of the clinical development is identifying which attributes are actually critical that define a product and go on to predict the clinical outcomes. So what exactly are CQAs? These are the chemical, or physical, or biological, or even microbiological attributes that can be both defined and measured to ensure that the final product remains within acceptable quality limits. Now, for all drug products, including cell therapies, CQAs are directly related to four things: the identity, the potency, the purity, and the safety of the drug product. Let's talk about each of these categories now. So first, identity. We need to have confidence that the product is what we say it is. And of course that sounds simple and straightforward, but specifically, we're required by law to have tests in place that can adequately identify it as the product designated on any final container. In the case of CAR T-cell products, the manufacturing process is designed to enrich specific T-cell populations, and the final product identity is often characterized by the presence of this chimeric antigen receptor, or CAR, on the surface of the cells. Protein expression is also used to identify other cells. For example, in hematopoietic stem cells, it's the absence of certain membrane proteins and the presence of others that identify these cells as hematopoietic stem cells. Now, a key challenge for identity testing of autologous cell therapies is distinguishing between one patient-specific lot and another. And while it would be possible to distinguish the cell lots by genetically testing each product, that's currently not practical. And so for these cell therapy products, careful tracking and labeling and segregation systems need to be in place in order to avoid mix-up between these patient-specific products. Next, let's talk about potency. Now, as with identity, we also need to have confidence that the product will produce the desired biological response. So that potency or power for cell therapies is determined by a number of different factors. Let's go over them now. First and foremost, we need to deliver living cells to the patient. Cell therapy is a living medicine. So we'll need to ensure that our product meets a target number of both total and living or viable cells. We also need a measure of the biological response. Now, we clearly cannot measure that in patients before administration, so we'll need ways to assess or correlate or predict with this in vitro. One potential measure, as mentioned earlier, is the expression of protein products made by the cells themselves. For CAR T-cell therapies, the number of cells expressing that CAR is another key measure of potency. And for hematopoietic stem cells, it's the expression of CD34. Therefore, some products will also need to deliver the correct number of cells that are both viable and express the desired proteins of interest. Another potential measure of potency is the ability of the cells to proliferate. In the case of hematopoietic stem cells, the ability of that cell type to proliferate is related then to the ability of those cells to differentiate into other blood cell types, as well as their capacity for self-renewal and the ability of those cells to engraft or remain resident in the patient. Finally, it may be necessary to measure the cell function in vitro related to how we think the biological response works. As an example, CAR T-cell therapies are designed to kill tumor cells in the body. So therefore, an assessment of their ability to kill tumor cells in vitro by measuring tumor cell lysis in the presence of those processed cells is also expected to correlate with the therapeutic efficacy in vivo. Now, despite their importance, potency assays are often some of the most difficult assays to develop. Why is that? Well, first, it requires knowledge of how the therapy works or the mechanism of action. That might not be well understood, and in fact, there might be multiple mechanisms of action. Second, potency assays are often biological in nature, as we just saw, and that makes them prone to high degrees of variability in testing. And finally, it may be difficult to develop a suitable reference standard to ensure that the assay is actually performing appropriately. How do we calibrate? Next, let's think about purity. Now, specifically, in our case, we want to ensure that the final product is free of unwanted impurities that may come from the raw materials or the manufacturing process itself. Some broad examples of impurities that might be added in the manufacturing process include residual solvents or antibiotics or animal products that might have been part of the additives to the cell culture media, or even unwanted cells that were part of the process in the starting cell material or added intentionally during the process. So for these types of components, we must either validate that our process removes them or test the final product to make sure that they're now below an acceptable level. It should be clear that these impurities are going to be both product and process specific. So let's take a look at a few specific examples now. In the case of CAR T-cell therapy, one of the key raw materials that's added to the process is that viral vector that we use to transduce the T cells and make them express the protein of interest in the first place. But therefore, the final product must be shown to have an absence of detectable viral vector. That is not part of the final product. Other types of cell therapies utilize feeder cells of animal origin, such as mouse 3T3 fibroblasts for the skin graft product Epicel. In this case, the final product is tested to ensure that those feeder cells are below an acceptable level. Now finally, let's talk about safety. In the context of biological therapies, and cell therapies in particular, when we discuss safety, we don't mean safety of the active ingredient. That part is evaluated during the clinical trials. But in this case, we need to ensure that there's no contamination of the drug product that would present a safety issue. Now, at its heart, safety is designed into the manufacturing process through approaches like aseptic processing and choosing the appropriate raw materials, like our media components and single-use plastics. We do that to minimize the risk to safety, and we test that process throughout to make sure that nothing went wrong. But let's go over some of those main risks and how we need to address ensuring that those safety risks are not present in the final product. First, let's talk about the absence of microorganisms that could present a risk to human health. Sometimes you'll hear this referred to as sterility testing. Bacteria and other microorganisms are some of the primary agents of disease in humans. So therefore, it makes sense that the final product has both been aseptically processed and then tested to ensure a lack of contamination of these microorganisms. This is especially important for cell therapy products because they're generally implanted or infused into the patient directly into the bloodstream or the tissue, and therefore they bypass some of the initial defenses we have against bacteria, such as our skin or the low pH of our stomach. We also need to ensure that our final product is free of endotoxins. Now, endotoxins are components of the cell wall of gram-negative bacteria, and the problem is that they induce an immune response in us. So having endotoxins in the final product could be an indication that there was bacteria in the culture, but it could be part of the raw materials that were used in the manufacturing, such as in the cell culture medium. So we need to ensure that the final product is tested to make sure that endotoxins are below an acceptable level, because if they're not, that immune response could include, for example, a fever. Next, there are requirements to ensure that the final product is free of a specific class of microorganisms known as Mycoplasma. Now, Mycoplasma are bacteria, but these are very small bacteria that lack a cell wall. So that makes those bacteria very squishy and deformable and able to get through the filters that would trap or keep other kinds of microorganisms and bacteria from going through into the final product. So since these Mycoplasma can evade our filters, and because they can be human pathogens, it's important to make sure that they're not in the final product and we test that they're below a detectable limit. Otherwise, these pathogens could actually come from the raw materials or the personnel or even the donor tissue and present a risk to health. The next category of microorganisms of concern is virus, such as Hepatitis C virus. Now, as we know, viruses are obligate molecular parasites. That means they co-opt the cellular machinery to produce more of themselves. So a key aspect of viruses is that they require a living cell host to replicate. And this makes it difficult for us to detect them in the absence of that host. So as with the other aspects of safety we've discussed, safety from the virus is largely built into the process itself. As an example, in the case of autologous cell therapies, the cells are taken from the patient and then given back to that same patient. And sometimes a patient's own cells may harbor a latent virus, for example from a past infection. So though those cells are going to go back into the same patient, there is a concern that the cell therapy manufacturing process itself may amplify that viral load in the cell product, and then when going back into the patient, negatively impact that patient. So therefore, it may be required to demonstrate that the manufacturing process itself won't replicate any latent virus that may be present. Now, for allogeneic therapy products, cells taken from healthy donors are used to manufacture the product. And here, the donor cells are subjected to extensive testing to ensure that they're free of contamination by virus and other adventitious agents before they're even used in the manufacturing process. A challenge for many safety tests, especially these that rely on the growth of the microorganism, is the time it takes to complete the test. In some cases, the shelf life of our cell therapy product requires that it be shipped to the administration site, like a hospital, before we even know the results of a particular safety test. We will discuss this challenge in more detail in later sections of this unit. Finally, in addition to the list of microorganisms, there are contaminants that can pose a safety risk, such as particulates from the manufacturing process or in the final product packaging. In the case of particulates, really just small particles, these are visually counted to ensure that they're below a target threshold in the final product. Now, before we go, we should note that final product release depends on more than the CQAs being within the target specifications. Final release is also dependent on an evaluation of how well the manufacturing process was controlled. And this evaluation uses in-process testing, batch records from the manufacturing process, and other measures of control, such as the raw material testing. As you can see, the critical quality attributes, or CQAs, of a cell therapy product can be quite complex. However, it's paramount to ensure that our product is what we think it is, will induce the desired biological response, is free from impurities, and is safe from pathogens. So throughout the rest of this unit, you will learn how specific assays are used to assess and define a product's CQAs.